Well, welcome everybody to another Science Unwrapped in our series for Brave New World. You know, it's an odd introduction when you say that you're sorry for a particular topic that you know is going to be exciting, but this is a case where we're both here with masks. I'm pulling my mask up. I'm sorry with how timely this topic is. With me right here is Dr. Brett Hurst, USU Institute for Antiviral Research. Brett will talk tonight about uneasy neighbors, so how interactions between humans and animals can cause pandemics. And I wish it weren't such an important topic for this time right now, but I don't need to tell you anything more about that. What I'd like to do is just, something I should have done a moment ago and introduce myself. My name is Greg Podgorski. I'm Associate Dean in the College of Science, but more importantly, I chair the Science Unwrapped Committee. And I want to thank everyone on the committee for making these events, especially in a challenging time, successful. So thank you to the committee. And along the ideas and lines of thank yous, I want to thank our volunteers who normally would be presenting to lots of school kids and parents and just having a great time after a Science Unwrapped. We can't do that this year. But we do have a lot of volunteers who have stepped up and have activities on our webpage. So please check out the Science Unwrapped after activities that are virtual this year, but are still really good. So we've got a lot of them up there, and the list continues to grow. So thank you for that, but please, those in the audience, please check those things out. We're going to look over here, just a few words about Brett. Brett is a Utah native. He earned his bachelor's at Weber State University in 2006. He immediately went to work at the Institute for Antiviral Research, where he has been since 2006. And along the way, Brett picked up his master's and PhD from the Animal Dairy and Veterinary Science of Utah State University. And you might guess of what Brett had done his graduate research on. And if you guess virology, you're absolutely right. So Brett has done work on both enteroviruses, not both, but enteroviruses, influenza virus, and more recently, coronaviruses. And he'll be talking about the later two viruses tonight. What we'll do is we will have a question and answer session following the talk. And this will be run through the chat feature, actually, I should say question and answer feature of the Zoom webinar. So as you have questions during Brett's talk, don't be afraid to send them in to the question and answer feature. And I will filter some of those questions, but then Brett and I will work together once his talk is over for a question and answer. And there should be lots of questions tonight. So without much more of an introduction, so Brett, I'll turn it over to you for your talk on uneasy neighbors. Okay. So thanks very much. Look forward to the talk, Brett.
Thank you, Dr. Podgorski, for that introduction. I'm excited and grateful for the opportunity to be able to be here to present tonight and to talk about how viruses and animals can lead to pandemics in people. So if you were paying attention at the beginning of this pandemic, if you were paying attention, you may have heard on the news that COVID-19 actually came from China, that it was released from a laboratory and has been spreading across the world. And so early news reports indicated that this, you may have even heard it referred to as a China virus. And so we're going to talk about that today and why that is and really where did this virus come from. Since that initial news report, scientists have actually determined that a lab release was unlikely. That's not the most likely culprit for this virus for where it came from. And that this virus most likely came from animals. And so tonight, that's what we're going to talk about. We're going to talk about where do viruses come from and how do we understand how they come from animals. To use that, we're going to talk about a couple of viral questions here. So the first one is, what is a virus? And we're going to compare a virus to a bacteria so that you understand kind of the difference, where you may be more familiar with a bacteria than you are with a virus. The next thing we're going to talk about is how do viruses from animals infect people? And we're going to use two different viruses to look at that. The first one is influenza, or the flu virus, as you've often heard it called. And the reason I'm going to start there is partly because I know a lot about that virus, but because we in general know a lot about that virus. The coronavirus, I know it seems like it's been around much longer. The current coronavirus has only been around a little less than a year. And so we don't know everything about it. We're still learning a lot as scientists and as people. And so we're going to use influenza as a model to determine where these viruses come from, and then we'll look at the current coronavirus. And then the last thing we're going to talk about is how does this create a pandemic? Or what is a pandemic? A pandemic is a disease that affects the whole world. And so how does this virus that comes from an animal, how does that spread throughout the entire world? So first, let's talk about what is a virus. So a virus is a microscopic parasite that is unable to reproduce outside of a host cell. And so this is a parasite that requires being able to get inside of our cells. It's made up of a protein coat and genetic material, which is either RNA or DNA, depending on the type of virus. You can see the picture here on the side. That is a picture of the coronavirus. And this is a picture of the outside of that coronavirus that's covered in that protein coat. And the genetic material would be inside. When viruses were first discovered, they were first viewed as poisons and then as living organisms because scientists couldn't see them and they didn't know what was causing the disease in people. And so they thought they were poisons, and then later they thought that they were actually alive. Now, viruses actually occupy a gray area between the living and non-living, or something that would be approximate to the undead. Because they can't reproduce on their own, they're not really alive. But they also can make copies of themselves and spread throughout the world, so they're not really dead either. They're kind of somewhere in the middle. When we compare viruses to bacteria, viruses are much more simple. You can see over here is a picture of a virus, and the yellow squiggly lines in the middle, that would be the genome, the DNA or the RNA inside of that virus. The pink part around it is that would be the capsid. The capsid is made up of proteins that help protect that genetic material inside the virus. And then this blue part around the outside is an envelope. That envelope can be on some viruses, but it's not on all of them. And then when you compare that to a bacterial cell, that is much more similar to our body cells, our animal cells. The next thing about viruses is that viruses cannot reproduce without a host. Well, when I say a host, what do I mean? The virus has to infect a cell. It has to get inside a cell to be able to make copies of itself, or replicate as we call it. And so here's a picture of a virus. You can see how it has to attach to the cell here. This would be any cell, whatever cell it's trying to infect. If in the case of the coronavirus, we are the host. And so that virus has to get inside our cells to be able to infect us and to be able to make more copies. But it can also, certain viruses can also infect animals as well. So that virus will infect the cell, make copies, and then distribute those new copies throughout the body and to other people. Whereas bacteria are capable of making copies on their own. The next thing about viruses and bacteria is that most viruses are bad for us. Most of them cause disease. And so if we get a virus, chances are it's going to make you sick. You've probably had a number of viruses in the time that you've been alive. Whereas if we compare that to bacteria, most bacteria do not cause disease. They can cause disease, but most are actually helpful and are a natural part of our bodies. Viruses also usually target a specific tissue, so they like to stay within one part of the body. So for example, the influenza virus that we'll talk about tonight, it will primarily stay inside of the lungs, inside your respiratory system, where bacteria are often spread throughout our body. Now, if we're trying to treat these viruses or bacteria, there's different methods that we have to do that. The primary way that we protect ourselves from viruses is using either vaccines or antiviral drugs. And we use those to be able to treat those infections when we get them. For bacteria, you would be given an antibiotic. Those antibiotics specifically target bacteria, but do not work against viruses. However, there are some vaccines that we have created for bacteria as well. And then just looking at the size difference between these two. So on the left here, you can see this large picture of a bacterium. That is a normal bacterial cell. And you can see that over on this side are all the different sized viruses that there are. So there are some viruses that are very large. These are called giant viruses. And then there's some that are very tiny. And when we compare that to the coronavirus, this is the coronavirus that causes COVID-19. It's somewhere here in the middle. It's not the smallest virus, but it's not the largest either. So where did these viruses actually come from? Well, most of the diseases that we as humans get actually come from animals. So they start in animals and then are passed to people. And why is that? Well, that's because animals are everywhere. Animals are our food. They're in our movies. They're in our books. They're on the internet. And they're in our families. We have a lot of close ties to animals everywhere that we are. And so we have a lot of contact with those animals and a lot of ability to pass viruses back and forth between us and animals. Much like if someone in your family gets sick and you pass it, and if your sibling gets sick and they pass it to you, the same way your animal can get sick and pass it to you as well. So if we talk about where these viruses actually come from, if I told you the story of a dog or a wolf that bit a man and the man started to act weird, the first thing that you might think of is a werewolf. You'd think, oh, that's where this came from. Well, what if I add in the idea of viruses? And we add in viruses, and I told you that viruses help to explain the story of werewolves. So now, if we told you that, what if this dog actually isn't just a normal dog, but it's a dog that has a virus? And that dog now bites a human, and the person now starts to act or abnormal. And the actual story of this is the rabies virus. So this is a virus that is carried in animals and can be passed to people. And when early doctors and scientists did not know what was causing this person to act abnormally, they knew that there was a correlation between the dog bite and the person acting strange. And what they later determined is that the rabies virus was a way that explained the idea of both werewolves and vampires, where we're passing diseases from animals to people. And so even early doctors, without knowing what viruses were, knew that there was a link between the animals and the diseases that they could cause. So the reason that animals can cause disease is because animals are what's called a reservoir for viruses, and we'll talk about exactly what that means. So there's a bunch of different diseases that can be spread between animals and people, and these are called zoonotic diseases. And you can see right in the word zoonotic that we have the word zoo, meaning it's from animals. So much like animals are housed in the zoo, these viruses or diseases are housed in the animals. So influenza virus is the first virus that we're going to talk about tonight. It's normally found in birds, and it also spreads to other animals and people. And the interesting thing about influenza virus is that it normally does not make birds sick. And so the birds actually can have the virus, but it does not make them sick. They don't get a cough, they don't get a fever, they don't notice that they're infected at all. So the normal host for this disease is called the reservoir. So for influenza virus, you can see on this picture over here, there's a bunch of different influenza viruses that are normally found in birds, and they are called the reservoir for that virus. So from that reservoir, it has to be spread some way to another animal. So the next animal that goes through is called an intermediate host. And so you can see here that these birds are able to pass these viruses and spread them to all of these different animals. And they can pass them directly into those animals, and then you can see that there are actually links from those animals. And so these viruses that get passed from, for example, from a duck or a geese, some of these wild birds, it can be passed into our pigs. And then you can see that there's a link between pigs and people. And so that virus can get passed from birds to the pigs and then end up in people. And the people are what's termed either a dead-end host, meaning that the virus stops there. It either causes some disease, and either the person gets better or they get sick and die. But usually it does not spread from that. Every once in a while what will happen is that we will get a spread of that virus where we will continue to pass that virus back into people. And you can see that there's these three viruses over here, and these viruses continue to pass in people. They don't go back into the birds, they just continue to circulate in people. So what happens when we get sick with influenza virus? If you've ever had influenza virus, you know you get very sick. So it's a respiratory disease, meaning that it infects your lungs. You may also get a fever, a sore throat, a cough, and you could get very tired if you have influenza. It infects about 8% of the population each year. And so that's in just the United States, about 8% of people. So about 1 in 10, you have about a 1 in 10 chance or a 1 in 12 chance of getting infected with influenza each year. And that's responsible for anywhere between 56,000 deaths, 170,000 hospital visits, and an economic burden of $87 billion. And this is every year. So every year we're fighting this just with influenza viruses. And occasionally it becomes much worse than that. And what happens is we get a virus that we have never seen before in people. And that's what happened in 1918. We had what's called an influenza pandemic. Much like the coronavirus pandemic that we have now, this was an influenza virus that spread throughout millions of people. At that time, 500 million people in the world were infected. So about a third of the entire population of the world. 50 million people ended up dying from infections from this virus, and 675,000 of those people died in the United States alone. The biggest problem is we didn't have any vaccines or antibiotics to be able to treat those infections. But this was such a wide-reaching infection, you can see here on the pictures that we had doctors and nurses waiting with hospital beds to try and treat all the patients. The bottom picture is actually a picture from the Utah State Gymnasium that was called the Smart Gymnasium at that time. It was actually called the Utah Agricultural College. So in 1918, they actually turned it into a space to house the Army Training Corps to be able to try and treat all of these infections. And at that time, Old Main was actually turned into a hospital to be able to treat all of these infected people. So this virus had wide-reaching implications for how many people it was able to infect. Since then, we've had additional influenza pandemics over the past 100 years. So in 1957 and 58, we had the Asian flu, where 1.1 million people died in the world, and over 100,000 of those died in the United States. In 1968, we had the Hong Kong flu, which had 1 million deaths in the world and another 100,000 deaths in the United States. And more recently, in 2009, we had the swine flu. And this was not quite as big, but we still had almost 600,000 people in the world that died from these infections. And you're starting to see probably a pattern in these. You're seeing that, that all of these viruses are coming from Asia. So why is that the case? Why do viruses actually come from certain parts of the world and not others? Well, if we look at this map actually color codes the risk of getting viruses from animals. And so you can see that they've actually colored it depending on how many animals carry diseases that can be passed to people. And so down here in the yellow and the orange, we have 10 to 20 animal species in that part of the world that can pass diseases to humans. As we get up here into the red, we have 30 and 40 different animal species that can do that. And as we get up here into the purple and the pink, we have 60 or 70 different animal species that can pass those viruses to people. So one thing that we need is animals that have those diseases. The second thing that we need is a lot of people being exposed to those animals. China has both of those. You can see China over here on the edge where we would have that nice red band right through the middle of it. It has a lot of animals that carry diseases that we can get. And it also has a lot of people. And so there's a lot of people that are able to get that disease from the animal and pass it to each other. So when we talk about influenza virus, we always want to determine how risky is it? How dangerous is this virus? For our normal influenza virus, so for the viruses that are always circulating each year, you likely have gotten a flu shot at some point in your life. There's viruses that are just seasonal. That means they come back every year. In the northern hemisphere where we live, those viruses are usually here from about October through February or March. But that mortality rate is about 0.1%. That means that's how likely it is that if you got that infection that you could die from it. And it's a little more complicated, but we won't get into all of that. For the 1918 pandemic, the mortality rate was about 2.5%, so about 25 times more deadly than our normal seasonal flu. If we look at the 1957-58 pandemic, the mortality rate's about 7 times more deadly, and the 1968 pandemic, about 0.5% mortality rate. 2009, less than 0.5%, about the same area. So if it's not really that many people that are dying, if it's only 0.5%, how bad is that? Well, remember, just from the seasonal flu, where we only have 0.1% of people dying, just that virus causes an economic burden of $87 billion, and we have 50,000 people a year that die from that. So just that virus that seems very low, we still spend hundreds of millions of dollars developing vaccines and drugs to fight that virus. So even a normal seasonal influenza can be very serious. But what if we talk about another more deadly virus? So one of the viruses I want to introduce you to is an avian influenza virus. So an avian influenza virus is also called bird flu or highly pathogenic avian influenza virus. So avian is just another word for bird, and pathogenic just means that it causes disease. So this is a disease in birds that causes a lot of disease. This virus actually has a mutation and it allows it to spread throughout the body. Where we talked about normal influenza wants to stay in your lungs, this virus can actually spread to your brain and to other parts of your body, which makes it much more dangerous. The other interesting thing is where most birds do not get sick with a normal virus, With this virus, birds actually die from it. So if they get infected with this virus, the birds will die. And it has about a 60% mortality rate in people. That means if 5 people get that disease, about 3 of those will not survive. The good news, I guess, with this virus, if there is good news to be had about a virus, it's that it does not spread well from person to person. So if I got that virus, I would not be able to readily spread that to another person. It can happen, but it's much more difficult. And you can see pictures here of birds that have died. This was at an outbreak in Nigeria where all of these birds that were agricultural birds died because they were infected with this virus. So the other interesting thing about influenza viruses is that they're able to recombine or mix together. So the 2009 swine flu was actually caused by 2 viruses that recombined. We had a virus from a bird that mixed with a virus from a person, and they mixed together in a pig, because a pig can get infected with both bird viruses and human viruses. And what that created is a new virus that our bodies didn't recognize, and so it was able to spread readily throughout that population. So what if we imagine combining one of those viruses, a virus that can spread very well, with a very deadly avian virus? Now what we would have is the efficient spread of a very deadly virus. So because of this, scientists have been very concerned about how deadly this could be if this virus existed in the world, and if this was actually evolved. So the important thing to remember is that all of these viruses are coming from animals. And in 2019, the CDC actually put together a list of viruses or diseases that they were worried about in the United States that come from animals. And as you look down this list of diseases, you probably recognize a number of those. The one that I want to point out is at the very bottom here, emerging coronaviruses. And if I just said this, but the Centers for Disease Control actually put out this article in 2019. So in May of last year, they actually said coronaviruses are one of the diseases that we're worried about in people. We're worried about something passing from animals into people. So why were they actually worried about that, and why do we need to be concerned about that? Well, the first thing that I want to talk about with coronaviruses as we're switching from influenza to coronaviruses is that these are not new viruses. The specific virus that's infecting us now is new to us, but coronaviruses have existed for hundreds of years. And so the first 4 viruses right here, these are human coronaviruses. That's what that HCOV stands for, is human coronavirus. There's 4 of these, and these circulate throughout the human population every year. And so at some point in your life, you have likely been infected with one of these coronaviruses. The good news is that these ones did not make us very sick. We might get a slight cold, just like you have every other year during the winter, but it really did not make us there was not a lot of disease associated with these viruses. However, we do have 3 other viruses that are called pathogenic coronaviruses. And those are SARS, MERS, and SARS coronavirus 2. And so those are the 3 that we're going to talk about tonight. You can see over here on the picture that each one of these coronaviruses actually started in an animal. So this is the reservoir in this column. And then you can see this is the animal that is the intermediate host that it was passed to, and then they get transferred into people. And so we'll talk about these last 3 because these are the 3 that are the most important probably for what we're discussing. So the first one is SARS coronavirus, the original SARS virus that actually happened from an outbreak in 2002 and 2003. So SARS stands for Severe Acute Respiratory Syndrome Coronavirus, which is a mouthful, right? That's why they shortened it to SARS. But it just means that we get a very severe disease that's acute, meaning it comes on very quickly. So after you get infected, that you will get very sick after that. And then respiratory syndrome, it makes it difficult to breathe, or it attacks your lungs. And then it's just telling you that it's a coronavirus that causes that. So this virus was actually normally found in horseshoe bats. And so this is the normal reservoir for this virus. And these bats do not get sick when they have that virus. However, in 2002, that virus was passed from bats to civets, which is a mammal that is common in Southeast Asia. And from that, people were infected by exposure to these civets. And from that, we had about 8,000 people that were infected. Over almost 800 of those died. So about a 10% mortality rate, meaning about 1 in every 10 people that got the virus died from it. The good news for this virus, once again, if there is good news, is that there were no asymptomatic cases, meaning that if you had the virus, you felt sick. Whereas with this new coronavirus, you can have the virus and spread it to other people even when you don't feel sick. And so it means without symptoms, meaning you don't feel that you're sick at all. And so the good news with this virus is that they were able to tell when people were sick, and it made them much easier to stop the virus. The second outbreak of coronavirus that I want to talk about is from 2012, so not that long ago. This is the MERS coronavirus, or Middle Eastern Respiratory Syndrome coronavirus. And this just says that it came from the Middle Eastern countries and it again attacked your respiratory system or your lungs. The origin is still not fully understood, but we believe it came from bats. Probably not Batman, but at least from bats. And that dromedary camels were the secondary host. And so it was passed from bats to camels, and then people were infected by being very close to camels, in close contact with them. Since 2012, we've had about 2,500 people that have been infected with this virus, and over 800 of those have died. So about 1 in 3 of those people have been infected with that virus and then died. The good news with this virus is that it does not spread well from person to person, and so it's easier to slow down that spread of that virus. The last one is one that you're probably familiar with, the novel coronavirus from 2019, or SARS-CoV-2. So again, it's that severe acute respiratory syndrome coronavirus, and this is the second one, and so it's coronavirus 2. It actually emerged in late 2019, and you can see we don't know when this will end yet. Again, we believe that bats were the likely source, though we have not identified the specific bats that are responsible for this virus. The intermediate host may be a pangolin, which is a mammal in Asia, and so exposure to these mammals that people in Asia often eat has likely been the source for this virus, but that has not been confirmed yet. And you can tell that from people being exposed to that, we've now had to, that virus has spread throughout the world. I had to update these numbers again because we have, as of today, 53 million cases worldwide that we've had and 1.3 million deaths. And so you can tell that this virus is spreading very well and is also very dangerous. So when we talk about COVID-19, so SARS-CoV-2 is the virus that causes coronavirus disease 2019. And for COVID-19, you can see they just took parts of the word to make up the COVID-19 here. And the mortality is difficult to interpret because this pandemic is still ongoing. We can't tell how many people are infected. It's hard to know if everybody who has been infected. And then we don't know how many of those are actually sick or if they do not have symptoms at all. What we do know is that the death rate is higher than the influenza viruses that we normally have, that seasonal influenza. However, it seems to be less than SARS or MERS, the original SARS virus or the MERS virus. So I guess that is good news that it's not as deadly as those viruses. However, this virus does spread more efficiently than the previous coronaviruses, and that's because of the way that it gets into the cells. And so it actually has to use what's called a receptor on the outside of the cell. And I don't expect you to memorize it, but it's called the ACE2 or angiotensin converter enzyme 2 receptor. So basically there's a specific, think of it as a lock and key for that virus to be able to get inside the cell. So the receptor I would compare to the lock. That's what keeps things out of the cell that the cell doesn't want. But the virus happens to have the right key to be able to get into that cell. And so that's what allows it to get inside. And so this virus happens to have the right key to be able to get inside our cells and spread effectively. So when you get COVID-19, what happens? Well, if you first get exposed to it, they'll have an incubation period where you may not have any symptoms. That's usually 5 days, meaning that the virus is infected in you, but it's not causing any disease yet. The second part is a symptomatic phase where you get symptoms. You might get a fever, a cough, you might get tired, might get muscle weakness or diarrhea, and that usually lasts about a week. And then after that, hopefully you lead into the recovery phase. For about 80% of people, they get better after they get infected. So you may hear people who have been infected saying, it wasn't that bad. Well, that's because they're part of the 80% that get better. However, about 20%, they get worse, and they actually have severe breathing difficulties, and they may die from it. And the reason that this virus is a little bit different is because we talked about viruses liking to stay in one tissue. This virus happens to spread throughout the body. And I'm not going to list all of these, but this virus spreads throughout many different systems in the body and causes lots of different problems. And so you may hear a lot of different symptoms that are associated with coronavirus infection. So part of my work at Utah State is I work for the Institute for Antiviral Research, and I'm one of 5 investigators shown here on the screen who all have different areas of expertise. And my areas of expertise are in influenza and coronavirus. So what are we doing with coronavirus? Well, here at Utah State, we're actually growing the virus in our lab. So we actually grow cells on a little plastic plate, which you can see over here. So these are little plastic plates, and on the bottom of those plates, we have cells that are able to be infected with the virus. And what we will do is we will test different drugs to see if those drugs can protect the cells from infection. And by doing that, that's what's called a cell culture experiment. And so we actually mix the virus and cells together, put them on a plate, and then we incubate it to see if those drugs protect the cells. The other thing that we're doing at Utah State is we're trying to establish an animal model of infection. And so what we do is we use this to evaluate different drugs or antivirals as well as vaccines. And so to do that, we actually infect different animals with the virus to be able to see if that virus can cause disease in the animals. And to do that, we've actually taken, we've got mice that have that human receptor, so they have that human lock on the mouse cells inside the mouse. We have hamsters that also have that human receptor or that human lock on the cells. And then we're also looking at possibly seeing if we can change the virus to be able to infect mice or hamsters. Can we get it to adapt or to be able to cause disease without those receptors? And what we're looking for is animals that model the human disease. Do we see the same diseases in animals or the same symptoms in animals as we do in people? So far, we've tested thousands of compounds against this virus in cell culture. We've also infected some animals with this virus, and in a normal hamster without that specific lock to get into the cell, the hamsters get a little bit sick, but they get better. But when we put that specific lock in there, that ACE2 receptor into those hamsters, it actually becomes a much more severe disease, which is similar to what we see in a lot of people. And so over here, you can see actually a picture that was captured by 2 of our scientists up here at the Institute. And you can see that these are lung cells underneath a microscope. And the different colors are different things that we're looking at. So the virus is shown in red, the receptor or that lock on the cells is shown in green, and then the lung cells themselves are shown in blue. And this big black space here in the middle, that's the airway space between our lung cells. And so that virus, you can see, is actually coating all of the surface of that lung tissue in there. So the important thing is that we are still learning. We don't have all the answers. We're still trying to learn which drugs can protect those cells, especially in cell culture. We found a few drugs that work, and we're still learning as much about them as we can. The other thing that we're still trying to learn is what signs of disease can we see in those animals? Do we see similar things that we see in people? Do the animals get a fever? Do they get a cough? Do they not feel well? And it's also difficult because animals can't tell us how they feel, so it's a little bit harder to tell exactly what's happening. And then lastly, can we find drugs or vaccines to protect the animals? And with the end goal in mind that we're trying to treat people. We want to find a drug that works in people, but to do that, we usually have to determine if we can protect animals first. And if it can protect an animal, there's a good chance that we can try it in people and see if it will protect them. And so just to kind of wrap up, I wanted to say that this is not the end. Obviously, with how long this coronavirus pandemic has gone on, this is not the end of these zoonotic diseases. We will continue to have viruses that spread back and forth between animals and people. However, to end on a more positive note, this will end. There will be an end in sight for this pandemic and for others in the future. As we continue to fight viruses that are passed from animals to people, we will get past this pandemic and we will be able to overcome that. I think one of the amazing things that we've seen has been the collaboration between different researchers, between the scientists and the government, and between even different pharmaceutical companies. As scientists, sometimes when we learn things, we want to kind of hoard that knowledge to ourselves, and we don't want to share it because we want to get our paper out there, and we want to have kind of that knowledge to ourselves. But as we've worked through this pandemic for the past 10 months, we've been so impressed with the ability of different laboratories to share their knowledge, to share what they've learned. And I think that it's really a testament to this idea of a brave new world. If you'd asked me before the pandemic if I thought that would happen, that scientists and pharmaceutical companies and the government would all work together and they would all share data without fighting, I would have told you it was probably impossible. But I think it's been really an amazing kind of thing to witness that all of these people have been willing to work together for the good of all of us. And so while there will be more pandemic diseases in the future, as a society and as people, we will continue to overcome them and to thrive and survive. Thank you.
A few questions coming in. I'm going to step, in fact, I will stay out of the way and have you in the spotlight, Brett.
Okay.
But first question that's come in is, have you had any success with the compounds that you've tested against the coronavirus?
So yes, we have. We found a couple of drugs that have worked okay, at least in the cell culture systems that we've used. There's a number of companies that we've worked with that have found drugs that look very promising. Keeping in mind that the work that we do is really the very beginning of a drug. And so we're usually looking at it much before it would go into people, really giving it just a general idea of will these things work. But there's been a number of them that have worked. And interestingly, we've also found a number that didn't work. So when the news media was talking about like hydroxychloroquine, we had already tested that. And I could have told you that it probably didn't have a very good chance of working. And so we knew that probably as early as anyone that it had a minimal chance. But.
You tested bleach?
We actually do use bleach for some things. So we use a lot of bleach in our lab. Bleach is a good thing. It kills viruses very well, and it kills the coronavirus fantastic. However, we know, most of us know that bleach is not very good for us. And so, while it works great to clean our countertops in our kitchen, it's not very good for us in general. So there's a lot of things we can find that kill viruses, but the trick is killing the virus without hurting us. So.
Okay, thank you. Another question has come in. So do you foresee the more frequent evolution of more viruses like SARS-CoV-2 in the future?
So I think there will be likely that that list of viruses that the Centers for Disease Control mentioned. Yes, I think there will be a number of those that will come into play in the future. So the coronavirus, I don't think we've probably seen the last of the coronaviruses. I think it has been amazing how quickly they've established a vaccine for this virus, where we've just heard news, within the past week that they do have what looks like a promising vaccine that could be, here. And most vaccines take years to get to where they're put into people. So I think that's kind of an amazing thing that we've been able to overcome to get that rapidly.
Okay. Yeah, thank you. This is a question from Ethan. From Amy. So what's the purpose of compounds tested in cell culture? Is it like a medicine that could be administered to humans or just for use in labs?
So both. So we have a number that are given directly that we use as some of them that we know that are not very promising, that may not work for people, but they work very good in the specific things that we're testing them in. But the big thing is that we're trying to whittle down the millions of compounds that are out there. How can we just choose the few, and which ones are they going to try and put into people? So they can't put everything into people because it's so costly. It costs millions of dollars to develop these drugs. So we have to kind of whittle it down somehow. And so testing them in cell culture is kind of the first step in showing that a drug will work. And if it can protect cells, then maybe it can protect an animal. If it can protect animal, maybe it can protect people. That's kind of the idea, is that there's a progression there.
So another question is that if humans are hosts, how do scientists decide when a quarantine is needed?
And that one is, so a quarantine, that one is difficult. So the quarantine is actually usually not decided by scientists. That would usually be public health officials that would decide whether or not it's a big enough crisis to need a quarantine. And of course, the public health officials are trying to weigh the benefits of a quarantine against the risks of everybody being cooped up at home, people being kept away from their jobs and losing their income, the impact that it has on the economic factors. So there's a lot that goes into that, and the scientists would help weigh in on that. But usually the scientists are probably not the ones who are making that final decision.
Then another question is that I've heard that in the future we're at greater risk for zoonotic pandemics. Why is that? And what's the relationship to climate change?
That's a very good question. So, well, basically our world has gotten much smaller. So if you think about, 100 years ago, what it would take for you to cross from America to Europe and how long it would take you to spread those diseases. Well, one of the big problems we had with the coronavirus pandemic is that people who were sick in China in the morning could be in Los Angeles by, within 18 or 20 hours. And so where they would still infect that, where 100 years ago, that wasn't possible. So basically, our worlds got smaller because of the technology we have with travel, and also with the fact that we just have more people here. So when the 1918 pandemic, we had 1.5 million people in the world. Well, we have or 1.5 billion people. We're over 7 billion people in the world. More people expanding into more territories that were previously held by animals, and so we're just pushing those boundaries between animals and people.
Okay. Here is a question that whether the COVID-19 is expected to be recurring, such like the seasonal influenza.
That's a great question, and the biggest thing about that is we just don't know yet. So the science is not back on that. I think that's been kind of the neat thing about this pandemic is we've been able to see science almost in real time. When scientists are learning about something, I will read a paper, and usually within a couple of days, I'll have a family member asking me about that specific thing. It gets picked up on the news. It's very everybody's very excited about it. So I think it's we're learning a lot about it, but really we just don't know whether or not this will be back every year or whether it will, if we can get rid of it. The vaccine will help, but we don't know if we'll have long-lasting protection. My guess would be, just based on the initial data that we've seen, that it will probably be around for a while. So it may not be something that we can get rid of in a year and not have to deal with again.
Yeah, related, and on a vaccine-related note, here is the old-fashioned $64,000 question, but now $64 billion question these days. When should we expect a vaccine?
That's a great question. I think you'll hear it when it's coming, but I think there's being a lot of progress made. So from what I've heard, it's kind of they're looking in the spring is when they'll start to see vaccines that are available for everybody. And even with that, the distribution of that vaccine is going to be hard. How do you choose who gets the first sets of vaccines? Do you give it to the people who are the most at risk? Do you give it to doctors and nurses? Do you give it to the scientists working in the lab with it? Or do you give it to government officials? So I think there's going to be a lot of challenges there. But I think with the progress that's being made, we'll probably have something hopefully within the next couple months, that we won't be waiting too long. So
that's hopeful. I certainly hope that. Along the lines of easy questions, could you speak about the potential for long-term immunity against COVID-19?
Yeah, and that's one of the problems that we just don't know yet. And so the early reports is that people who have been infected, there have been some people who have been reinfected again, who have had a second recurrence of that virus. And we don't know all the details about what that entails. So I would expect over the next year or so, we'll start to learn what kind of long-term immunity that we have. But with the coronaviruses, those circulate every year, the normal seasonal coronaviruses. And we have not built up a strong immunity to those. So there might be something specifically with these viruses that does not allow our body to really build up that long-term immunity. Now, with a good vaccine, possibly that could change, but we still have to kind of figure that out and see if that's something that we can change. But I certainly hope that we can get to that point.
Yeah, and I'm kidding about the easy questions. These are all good, but very difficult questions. A lot of them, as you point out, without answers just yet. So still along the vaccine lines, so speaking of a vaccine coming out quickly, how much should we trust this vaccine to be helpful without being harmful?
Well, I think that's the good news, is that as we followed what's been happening in the world, the U.S. government has decided that they would take the more cautious route with this vaccine. And so they would be more cautious about making sure that it was safe before releasing it into people. The initial data that they've shown is that this vaccine does make you feel a little worse. If you've ever gotten a flu shot, usually you'll get that kind of pain in your arm. You know that you've got that shot, but usually within a day or so it's gone. And this may be slightly worse than that. It may be kind of like some of the more severe vaccines we have, where you get you might feel a little worse for a couple of days. But so far, they haven't had any real negative side effects from that vaccine. So I think it looks promising so far. But really, we're just going to have to see what the data says. But I have been encouraged that the government has been very much on the safe side of things, that they want to make sure that they're not going to make the situation worse.
This is a question, too, that is unrelated to the vaccine, but it's a perceptive one about biology of viruses. So do all humans have the ACE2 receptor, and would there be some protection if individuals didn't have the ACE2 receptor?
So that's a good question. So yes, all people would have that specific receptor. they're still doing research into the different susceptibility or how easy it is for a specific person to get infected with that disease. We know that some countries have just had lower rates of infection compared to other countries. For example, Italy was very hard hit by this virus, and we still don't know how all of the genetics of the people and the country play into that. But certainly there's some of these things, the receptor might be one of those things that plays into that. And that's a great question, and one that scientists are working feverishly on, and not one that I think they have a definitive answer for. But it is likely that if you did remove the receptor, it probably would not help, because we still need that receptor in our bodies. And so it would probably not make you immune to the virus, or if it did, you would have other problems. So,
a question that I'm paraphrasing is, could you tell us a bit about what types of vaccines are being developed or looking at for being developed?
Yes, so there's a number of different vaccines that they're actually looking at right now. A number of them are actually an mRNA vaccine. And so it's, which is a little bit more complicated, but basically instead of our normal vaccines that we're used to are like influenza vaccines. And is what we do for those influenza vaccines is we kill the virus, which is funny because they're not really alive, but we do kill them. We make sure that they can't infect any cells. And then we inject that virus into our body and let our body build up that immune response to be able to fight that infection. And so is what they're doing with this, the mRNA vaccine, the one that is probably the closest, they actually inject this mRNA into your body, and rather than an entire virus into your body, that mRNA is just making that protein. So it actually makes a surface protein on the virus so that your body can recognize that virus if it sees it again. So kind of the opposite of that lock, we're actually making copies of the key and saying, this is what the key looks like. If you see this key, don't let it in. And so that's kind of the way some of these vaccines are working. So they're working on, some of them are mRNA vaccines. Some of them, they're actually chopping the virus up into small parts. They're called a subunit vaccine. And then they take parts of that vaccine and put it into people. So there's a number of different strategies that they're working. And they're trying to find out which one is the best. Does one of them work better than the others?
Okay, yeah, thank you. So is it true that your blood type can affect how you react to SARS-CoV-2?
I haven't seen that one exactly, but I know, but SARS-CoV-2 does create problems in the blood, and that's one of the things that this virus kind of gets everywhere. And so some of the problems that they found have been problems with blood clotting. So people that have been that have been infected with SARS-CoV-2, they've had like an increased risk of having a stroke. And so the blood type could obviously play into that. And I don't think that we know all the answers of how that works out yet. But yes, that could definitely play a role in that.
There are no other questions that have come in. So I'll give the gone call. So we're out there. So what I'm going to do is come around, Brett. I will thank you. I'll give you a virtual clap over here. So
thank you.
So thanks for a great talk. And I want to put in a thank you too for the USU media production team who really make this possible for us. So thanks very much. And I'm going to put in a plug as well for the Institute for Antiviral Research, which is just such an amazing facility on this campus. And I really have been just, again, incredulous with the kind of work that you do and others do there. So thank you.